Good morning, Manchika. So we have a new worksheet today with more than 100 questions. 143 questions are there. This comprises of NEET questions from 1988 to 2016, previous year NEET papers. So many of this we had solved. Good morning, Tarun, in the last class. So I'm Rahul Saha and today we'll be solving the questions that we hadn't solved in the last worksheet, because the last worksheet was about 2011 to 2019. This one is from 1988 to 2017. Good evening. Good morning, Ayushi. Ayushi, regarding biotech class, like when do you want to have it? Good morning, Priyanshi. The biotech students, like we were, yeah. Good morning, Ayushi. So when do you want to have biotech class? Like on Sunday evening or Monday evening? Like what will be comfortable for you regarding the biotech topics? Okay, okay, Monday evening. Okay, fine. Okay. So let's start with the question number. Let's come to, in that worksheet, come to question number, because the first few questions we had solved. So come to question number 16 directly. Till 15th we had solved only. So come to question number 16. So Balbiani rings are sites of option A polysaccharide synthesis, option B RNA and protein synthesis, option C lipid synthesis, option D nucleotide synthesis. Okay. Good morning, Ayman. So Balbiani rings are nothing but, I'll tell you what they are. They are present in polytene chromosomes. So polytene chromosomes are present in Drosophila melanogaster. They are a specific type of chromosomes. There is asked in NEET examination. From that part only this question is has been presented to you. So polytene chromosomes, they occur in Drosophila. We have talked about it a lot, fruit fly, in chapter number 5, Principles of Inheritance and Variation, Genetics chapter. So what happens is they are produced by endoreplication theory, okay, or by simply known as endoreplication. Means after replication there is no segregation of chromosomes. They keep on getting produced, they keep on getting produced. What happens is 10 rounds of endoreplication happens during the S phase. Okay, there is no segregation of chromosome in the M phase. That is the thing. In S phase the chromosomes, they keep on replicating. So you have 10 rounds of replication, or you can say 10 rounds of endoreplication. They keep on replicating without getting segregated, right? So 2 DNA strands to the power 10. How many will you get? 1024 DNA strands. This is the sheer number of DNA strands that are arranged alongside each other that produce distinctive banding patterns. Okay, that is known as endoreplication. Okay, and these are the sites. Okay, they are large puff sites. Okay, Balbiani ring are what? They are large puff sites in case of polytene chromosome. Okay, they are present during the larval developmental stage. Okay, of the dipterians. I'll just show you what are Balbiani rings and polytene chromosome for you to understand. So Balbiani, they look like puff. This is how they look. So these are the sites of, you can say, Balbiani ring. We have NOR sites also, nucleolar organizing region. Okay, so you can see this question coming here also. This is previous year NEET question. You can see this stuff. These are the Balbiani rings, the puffed up rings in polytene chromosomes. These are the, you can say, Balbiani ring. Okay, you can say BR, Balbiani ring. Okay, these are the Balbiani rings. Keep on, you can say, replicating. That is the polytene chromosome. So Balbiani rings are the puffed up sites. Okay, inflated sites, you can say, on polytene chromosome only. Okay. So, and these sites are extremely euchromatic in nature. Euchromatic in the sense they are very less bound or very lightly bound to histone, and thereby they are an active site of, you can say, option number. You can tick here option number, question number 16. Okay. So let's check the options. RNA and protein synthesis, polysaccharide synthesis. So it is never polysaccharide here. So it is B, RNA and protein synthesis, and between RNA, that is mostly RNA synthesis, because once your mRNA is synthesized, then only your protein will be synthesized later on in the rough ER, in the rough endoplasmic reticulum, containing the ribosome. So answer option number B is correct here, because it is a puffed up site, Balbiani ring, okay, which causes mRNA synthesis. It is mostly euchromatin. Euchromatin, so high transcription rate or high expression rate, you can say, expression and transcription, that is possible. So you might have heard of NEET examination result this time. So two students, they scored for the first time in the history, one boy and one girl, okay, Anshika and Aftab. They scored 720 out of 720 full marks, complete full. Like not a single question did they do wrong, not a single question did they leave. Full marks for the first time. So that's a great achievement by them. Then we had done question number 17. We had done question number 18 also. We had done question number 19 also. Okay, 20 also was done. Okay, 21 also was done. 22 was done. Then 23 was also done. Now come to question number 24. Okay, that's a good question. Question number 24. Now in question number 24, which of the following statements is not true of two genes that show 50% recombination frequency? Okay, the recombination frequency must be 50%. Okay, then which is not true? The genes show independent assortment. If the genes are present on the same chromosome, they undergo more than one crossover in every meiosis. Okay, the genes may be on different chromosomes. The genes are tightly linked, which is not true. Very, very important. So not true here will be they are not tightly linked. If they were tightly linked, they would not undergo 50% recombination. They would be undergoing very less recombination, because I had explained to you many times that linkage is inversely proportional to, you can say, recombination. Or I can write it like this: 1 upon recombination. So it's an inverse relationship always. So if you have got two genes, two chromosomes only like this, if they are pretty close by, okay, if you have if they are far apart, then their recombination frequency will increase. So that is what has happened here. Okay, that is what has happened here. So I told you that also, that concept, that even if you go for 100% recombination, the total recombination will still be 50%. Okay, because these two distal chromosomes, they'll never take part. Okay, they'll never take part in recombination. So if you have a single crossover, I'm drawing a single crossover only. DCO I had also shown you. Double crossover products, we will be having this only. A, B, here you have got crossing over done. So capital A with, it goes with small b here. Here it remains small a, small b, parental type. Here small a goes with capital B. So only here you can say, you can see even if 100% recombination happens, you will see only 50% of the DNA strands being recombined, because the distal ones, chromosomes that do not undergo recombination, they will show parental type, small a, small b, capital A, capital B. So only out of 4, these two, this one and this one, these two are the recombinants. Okay, or single crossover products, SCO. Okay, these are the parental type. Okay, parental, or I can also call them NCO, non-crossovers. Okay, so that's the concept behind this question. Coming to question number 25, the diagram shows an important concept in the genetic implication of DNA. Fill in the blanks A to C. Very easy question. C. Easy questions are also asked. Okay. So DNA to mRNA, what is it? That is transcription. mRNA to protein, translation. Okay. And then you can say protein to C. Okay, A, B, C. So protein, it was again proposed by, you can say, not Rosalind Franklin, not James Watson, but you can say Francis Irving Chargaff. He proposed the Chargaff rule. So it was proposed by Francis Crick. So 25th option A would be correct. Very easy question. Okay. Now 25 done. Question number 26. Which enzyme will be produced in a cell if there is a nonsense mutation in the LAC-Y gene? Very, you can say one of the tough questions, but it is asked in NEET examination a lot. Okay. Question number 26. If you come to question number 26, trans-acetylase, nonsense mutation in, you can say, LAC-Y gene. Okay, that is the question here. Just think about it. LAC-Y codes for what? It codes for the ZYA permease, right? So just think what would happen if there is a nonsense mutation. Nonsense means stop codon. You are not able to produce any more, you can say, LAC-Y, or LAC-Y will not be produced. That is the concept here. Just think what it could be. Just try to give it a shot. Once you start thinking, you will be able to solve little bit. Okay. Question number 26. So the option C here is not J, that is beta galactosidase. Okay, that is beta galactosidase, not J. Okay. So by, you can say, technical printing error, that is J galactosidase. Don't think it is J, it is beta. Okay. Okay, so let me describe it to you. So nonsense mutation is what? Is the one which will stop the polypeptide synthesis due to the formation of termination or nonsense codon. This much you know. Now in lac operon, we have a sequence of structural genes ZYA, which I have described to you many times. Okay. If I draw like this, negative inducible operon, lac operon, okay, you have Z, you have Y, you have A. You have the operator region, promoter region, the cap site which we had talked about in the last class. Then you have LAC-IN, promoter for LAC-I also. This is your LAC operon concept. You will be having a polyhistronic mRNA comprising of these three genes. Now Z codes for what? Z codes for beta galactosidase, Y codes for lactose permease and thiogalactoside transacetylase. Okay. Lactose permease, thiogalactoside, okay, transacetylase, or simply transacetylase you can also call it. Okay. Now, if the gene Y has a nonsense mutation, gene expression will stop at it. Okay. Gene expression, if it stops, that means what? Both gene and successive gene A will also be stopped or not? Because if I am saying nonsense mutation in Y, so will A be transcribed? Not at all. If you have UA, UGA, UAG here only, if there is a nonsense codon / stop codon at Y only, you will never get mRNA. mRNA will start from where? It will only be till here. And this is half, so this is gone case. Only entire structural gene of Y will be present. You will not be able to go till A, even if A doesn't have any kind of nonsense mutation, because we have terminated the transcription, because it started from the promoter region, which is upstream of Y and upstream of Z. So there is no way that you can make your mRNA transcribe the A gene. So now come back to the question. Okay, come back to the question. Which enzyme will be produced? So there is no way that lactose permease can be produced. Okay, because it has got, you can say, nonsense mutation. Transacetylase, it cannot be produced, and B, again lactose permease transacetylase cannot be produced. Which can be produced? Only the product of the Z gene. That can only be produced, because that is upstream of Y. So the answer will be C, that is beta galactosidase. Don't worry, this is a spelling, this is a PDF, you can say, error. That is written J. So option will be C, beta galactosidase. Okay. Always remember, option A is only this one will be synthesized in case you have a nonsense mutation there. Okay. So that is how you solve these kinds of questions. Okay, nonsense, missense, many things can be asked in lac operon. Okay, so that is how you are able to solve these questions. So if you solve even more, okay, I can make you solve 2-3 more questions on lac operon only, just for your practice. Okay. So if you take LAC-I, okay, or you can say lac operon expression model. So if I say the repressor of the lac operon, okay, or inducer of the lac operon, let's just explore lac operon. Okay, so inducer of lac operon, repressor of lac operon, we can start, you can say, solving questions like this. So let's start with the operator region and inducer region. Okay, so we all know that the lac operon is negatively induced. Okay, so here what I want you to know is, suppose the operator region, okay, the O region here, if I change the question, if I say operator region has a mutation which cannot bind, okay, which cannot bind, operator region has such a mutation, okay, operator region has such a mutation which cannot bind to the lac-I repressor, what will that result in? That can also be asked. What will that result in? Okay, so that will result in, you can say, always constitutive expression of lac operon, because there will be no inhibition, right? So it will be an constitutive expression, I can say. Constitutive means always. Okay, constitutive expression of lac operon, because lac I can never bind, so it will never be able to, you can say, inhibit the transcription. Okay, and if you talk about lac-I locus, okay, so in lac-I locus, if you have a mutation, okay, if I cause a mutation in lac-I, which is known as lac-I minus, lac-I minus means it is such a mutation which will produce repressor, okay, which will produce a repressor protein lac-I, but the repressor protein cannot bind to the operator region, or the repressor protein will not be produced. It may have a missense mutation which will reverse its configuration, or it may have two things here. I'll erase all this, okay, so that the concept remains clear. Lac-I minus, if the question comes in NEET examination, okay, this question has, these kinds of questions are asked mostly, okay. So lac-I means you can have two things. Either you can have a missense mutation, in a sense that it cannot bind to the operator region. You can have a nonsense mutation also, because the kind of mutation I haven't told. I just told minus means negative. Negative mutation could be missense, could be nonsense. Nonsense could be no lac-I produced. Okay, here produced, but it's incapable of binding to the operon, to the operator region of the lac operon, because of conformational changes, and this one is incapable of binding. In both the cases, what will happen? Again, since this operator region is vacant now, no operator can bind, sorry, no lac repressor can bind, again the lac operon will have constitutive expression, means it will always be expressed, it will not be constitutive expression, it will never be under any kind of repression or any kind of silencing, always expressed. Okay, so these kinds of questions are very helpful when you try to solve any kind of lac operon question. Okay, so one is this one, I told you, this is known as OC mutation. OC mutation, if you see NEET examination, that will preclude the repression. Okay, will not recognize the repression, and okay, the lac Z, Y and A will be expressed whether or not, you can say, lactose is present, always it will be expressed, because there is no repressor, there is no need to inactivate the repressor at all. Okay, and one more repressor I'll tell you, OC and I, they'll both make constitutive. Okay, one more I'll tell you for your NEET examination. If you're dealing with lac, let's finish lac here only. So one type of mutation in lac-I gene is known as lac-IS. If you have this kind of mutation, this is known as a super suppressor mutation. super suppressor means it is such a mutation, okay, which will prevent, okay, the repressor polypeptide from binding lactose, and thus will be, it is such a mutation. Just listen to it one more time. The mutation which made it IS doesn't let LAC-I protein or repressor protein to bind lactose. That is the meaning of IS mutation. The meaning of I minus was it cannot bind to the operator region, but here it cannot bind lactose. If it cannot bind lactose, then LAC-I will be always bound to the operator region. It will always bound to the operator region, and then it will be a non-inducible. It cannot be induced by beta 1,4-allolactose. Its binding will be, you can say, irreversible. So it will be, this operon will be what? Constitutively repressed instead of becoming constitutively expressed. In case of LAC-I minus and OC mutations, if this mutation is present, it will lead to constitutive repression. Constitutive repression of LAC operon. Okay, because it will bind to the operator region, and beta 1,4-allolactose cannot bind because it has got such a mutation, so it can never be unbound from the operator, and no ZYA can be produced. Constitutive repression, that is what is known as a super suppressor mutation. S for super suppression means it cannot bind lactose and can bind operator very tightly, but can bind operator tightly. That is the meaning of super suppressor here. So 3 questions like this can also come in NEET examination from LAC operon regulation. Okay. So now coming to our next question. So question number 27 is just the opposite of that only. So answer number D will be correct for question number 27. Come to question number 28. The satellite RNAs are present in some. Question number 28. Okay, they're not present in viroids. They're not present in prions. The prions are what? Prions are extremely, you can say, infectious proteins that do not contain any kind of RNA or DNA. Okay, viroids or virusoids are made up of RNA without any protein coat. They are just the opposite. Okay, viroids. You have virusoids also, because this is also asked in NEET examination. You have prions also. Okay. So what are prions? Prions are nothing but they are infectious proteins. Okay. They are misfolded infectious proteins. That's all. Misfolded infectious proteins which can cause disease. Okay, that is prion for you. So it can cause diseases animals and humans. Okay, by triggering normal healthy. Okay, by triggering, you can say, normal healthy proteins in the brain to fold abnormally. Okay, so that can be done by prion. Okay. So you have mad cow disease, okay, that happens in, you can say, a prion by, or infected, suppose if a cow is infected by a prion, okay, you will be having mad cow disease, and technically that disease is known as bovine spongiform encephalopathy, okay, BSE, spongiform encephalopathy. This question is also asked. Encephalopathy. Encephalon means brain. Okay. Brain degradation. Pathy means dysfunction. Okay. So that is mad cow disease by prion. Okay. When you look at viroids, okay. So viroids are the smallest infectious pathogens which are known. Okay, they contain a short strand of circular single-stranded RNA which has no protein coding. Okay, circular RNA, circular ssRNA. Smallest, one more question can be framed here. Smallest infectious particle is viroids. So here the tertiary structure of RNA, it provides it the stability, but there is no protein coating. Okay, no protein, no envelope, no protein coat. Okay, whatsoever. It is naked nucleic acid, you can say. Okay, and bacteriophage, we all know what bacteriophages are. They are the viruses which feed on bacteria. We have CRISPR-Cas9 from there. We have the generation of restriction enzymes in bacteria as an immune response. So the correct answer of 28th question, okay, satellite DNA is present in eukaryotes only. So it must be in 28. The trick is in eukaryotes. So it must be your option number D, which is plant viruses. Okay. Now come to question number 29. Which of the following is not a property of the genetic code? Very easy. Okay, question number 29. That is ambiguous. Genetic code is unambiguous. Okay. Genetic code is not ambiguous. Now, what do you mean by ambiguous? Okay. So, unambiguous means each codon codes for just one amino acid. Okay. One codon cannot code for, you can say, more than, you can say, one amino acid. That is the, each codon only codes for just one amino acid. That is the thing. And most amino acids are coded by more than one codon. That is degeneracy. But one codon is always coded by one amino acid. One codon, suppose AUG cannot code for any other amino acid other than methionine. Any other amino acid, any other codon can code, but except methionine and tryptophan, because these two codons are coded by only one codon. But suppose you talk about serines, we have 6 codons that can code for serine. That is completely fine. But one codon, if you take one triplet codon, that can never code, or that is never able to code for more than one amino acid. So that is known as unambiguity. Okay, it is not ambiguous that it will keep on coding for multiple amino acids via one codon only. Okay. So, we come to question number 30. Genes of interest can be selected from a genomic library by using, if you have a genomic library, you must be using DNA probe. Okay. So the answer will be here DNA probe. And what is DNA probe? In molecular biology, a hybridization probe. Okay, question number 30 I am talking about. In molecular biology, a hybridization probe is a fragment. Okay, it is a fragment of DNA or RNA. Okay, DNA or RNA of variable length which can be labeled radioactively or with a fluorescent dye, radio labeled, or you can say fluorescently, or fluorescent, or you can say fluorescently labeled in order to view it, right? Then it can be used in DNA or RNA samples to detect the presence of, you can say, nucleotide sequences which I wanted to detect. Okay, so that can be done. So those come under nucleoside or nucleic acid probing. Okay, so suppose you have a DNA molecule here. Okay. So it is nothing but a very short sequence. Okay, a short sequence of nucleotide bases that will get bound to specific regions, just like microRNA binds to the 3' UTR via its seed sequence, okay, which is of 8 nucleotides, okay. So, and degree of homology between the target and probe results in stable hybridization, okay. So, the range could be, if you talk about a probe, so the range of a probe could be as short as 10 bases or 10 nucleotides, okay, or it can also be as long as 10,000 base pair, okay, that is an exception, okay. The most common size ranges from 14 to 40 bases, that is the most common, very short sequences, okay. So suppose you have this DNA and you want to know whether I have this specific region in my DNA or not, so I'll put a probe which must be complementary in sequence, 14 to 40 base pairs, and if it gets base paired and this region is fluorescently labeled, and then I isolate and check the sequence, what does it mean? That sequence which I thought it would be present is actually present in the DNA. That is how probe works, and that is how I can isolate, you can say, in that question. Genes of interest can be, I can select genes of gene of interest, I can sequence it from the genome library, okay. That is what happens. So we have short probe, long probe, we have nucleic acid probe which I'll be using here, okay. Then labeling method is there, okay, that is not in your course, that will be in the class 12 biotech course. We have NEET translation, you have primer extension, okay, end labeling. So those things are not there in your course, in biology course, but will be there in the biotechnology course. So we'll move on to the next question, okay. So coming to the next question, in an inducible operon, very important thing, inducible operon, the genes are usually not expressed unless a signal turns them on. Question number 31 I'm talking about, okay. Then option B, usually expressed unless a signal turns them off, never expressed, always expressed. 4 options we have, okay, in an inducible operon. That's what we want to check here, inducible operon. So I have already described to you, or what is inducible operon, okay, what is negative inducible operon, right? So if you remember correctly, I'll draw the diagram again for you. Inducible operon and non-inducible operon. Okay. If we talk about inducible operon, that means what? You need to have a signal, right? Without a signal, it cannot be, or the gene can never be expressed. The gene can never be expressed because this comes under the gene regulation system, right? And under gene regulation, there are many types of operons, okay. You have lac operon, you have trip operon, but that is not in your course. But in an operon, I'll just draw a very good diagram for you where you will understand what do you mean by an inducible operon and what do I mean by a non-inducible operon, okay. So just draw this diagram, keep it with you for life, okay. This will solve all your problems regarding inducible and non-inducible type. Now I can put here negative control on top, a simple diagram, a very conceptual diagram. Then I can put positive control here. Okay. Then I can write here induction. I can write here repression. Now see, you have a repressor, okay? Negative control means what? You have a repressor like I, which will bind, so you have active repressor, okay? First of all, the repressor will be active, and this active repressor will switch off the expression or transcription. T means transcription, will switch it off. Okay. Now when I'll add the inducer is beta 1,4-allolactose, okay, or which is IPTG, okay, in case of the blue white screening, okay. So inducer, it does what? It binds to the repressor, active repressor plus inducer, and we make it, you can say, inactive repressor. Inactive repressor. Okay, so which option will be correct here? Option A will be correct. Usually not expressed unless a signal turns them on. So the signal is inducer here. That is known as inducible operon. You need someone to induce something. Otherwise it will never be induced. Okay. Now explore the other things also. What is positive control? That is why lac operon is a negative inducible operon. See, false and negative inducible. So negative inducible. Now positive inducible will be how? How will you have positive inducible approach? Okay. If you have an inactive activator, activators, they recruit RNA. Okay. If you have an inactive activator which will recruit RNA polymerase, inactive activator cannot recruit RNA polymerization, or sorry, transcription cannot begin. And now when you have an inducer which makes inactive activator an active activator. So here I am talking about activator, not repressor. So it is a positive control. Now mRNA synthesis can happen because RNA polymerase enzyme can be recruited. That is positive control. Now coming to negative repression. If you have an inactive repressor, okay, means the operon is on and you have inactive repressor. But then a co-repressor comes which makes this inactive repressor active repressor. And shuts down the operon. Now operon off. In the beginning operon was on, now operon is off. That is negative repression, because repressor was inactive in the beginning, okay, and co-repressor made it active. Just the opposite of, you can say, inducible. And positive repression means activator was active, okay. Active activator means again here operon on, but co-repressor will come and bind, cause conformational change, and will make it inactive activator which cannot recruit RNA polymerase. Therefore no mRNA, therefore operon off. So here you are repressing means operon is off. Sorry, operon is on in the beginning, then you are switching operon off. But in case of inducible, operon must be off, you should induce it to become on, just the opposite. So there are these 4 levels of control of any operon. Okay, so lack operon falls here. Other operons are not in your course. Okay, lack operon, I can only give the example of lack operon, which falls under negative control. Okay, negative inducible control. So now coming to question number 32. One of the most frequently used techniques in DNA fingerprinting is 32. Okay, very easy. A, VNTR, variable number tandem repeats. That's what we try to find via RFPD, RFLP, restriction fragment length polymorphism, or randomly amplified polymorphic DNA, or amplified fragment length polymorphism, AFLP. Okay, VNTRs, right? Then we can have satellite regions. We can have, you can say, mini satellite region. Then we will be having the microsatellite region. All I have described to you in detail. Okay, question number 32 answer is 1. Now question number 33, removal of introns. Okay. And in question number 28, if you want to know more, okay, about plant viruses, so the satellite RNA, you can say in case of plant viruses, are almost 194 to 1500 nucleotides in length. Okay, and the larger satellite RNAs, or the larger satellite, you can say, DNAs, they contain open reading frame and also express proteins. Okay, and the smaller satellite regions, they do not produce functional proteins. So that is an added info for you in case of question number 28. Okay. Now question number 33. Okay. So 33, yes. Removal of introns and joining of exons in a defined order during transcription is known as looping, splicing. Okay, must be question number 33 D, that is known as splicing. Okay, I have multiple times I have told you that splicing is an event which happens concomitantly or simultaneously along with the transcription event in case of eukaryotes, because eukaryotes, in many of the genes you have intragenic regions. Suppose this is exon 1, intron 1, exon 2, intron 2. So in last class also we had talked about it, that is intragenic, you can say, non-coding region, that is intron. So must be spliced out. Okay, and that will provide you the way of alternative splicing. You can produce more than one protein from one gene. So one protein, one gene hypothesis is completely wrong. This I had described to you in detail in the last class, so I will not go in detail in today's class. Just know that you have type 1 introns, type 2 introns, but those are not in your course. Okay, so in certain cases you have self-splicing introns also. Okay, type 1 introns. Okay, but you have spliceosome, ATP requiring introns. Okay, spliceosome complex, that is known as, it is the most important. You have multiple, you can say, these are the small nuclear RNAs and they are associated with proteins, so they become snRNP, small nucleolar ribonucleoproteins, okay, which produce a spliceosome, which was there in your question, which we had solved last time. So it is again present in eukaryotes. Okay, these are not self-splicing, but we have self-splicing introns also, type 1. Okay. So coming to the next question. If one strand of DNA, okay, has the nitrogenous base sequence at ATCTG, what will be the complementary RNA strand sequence? Complementary RNA that they have asked. Okay, complementary RNA. 34. Okay. So must be what? ATC TG. Okay. The complementary RNA must be UAGAC. That will be the complementary, it is called RNA strand sequence. Because complementary means this is the template strand I am talking about. Okay, this is the template strand I am talking about. ATC TG. So template means non-coding strand, minus strand, antisense strand. Okay. A to T, A, G, A, C. Now RNA which is coded, this is the complement of this one, but it stays in the 5 to 3 prime direction. So it will be U, A, G, okay, and A, C. UAGAC. Answer will be D. Okay. Then coming on to question number 35, very important questions. Ribosomal RNA is actively synthesized in the nucleolus. So rRNA which is present at the highest abundance inside the cell, okay, it is produced in the highest concentration. It is produced in the nucleolus. Okay, it is produced in nucleolus. Always remember rRNA. And nucleolus is a specific nuclear body. You have many other. You have this. You can have many other nuclear, or you can say nuclear bodies. Kajal body, then nucleolus, okay, and many other could be also present. Okay, promyelocytic bodies are present, so those could be present, but nucleolus is one of the most predominant regions. Okay, it is associated with, you can say, it contains nucleolar organizing center also, nor you can have 10 nuclei per cell. Okay, that is again a concept, and each nucleolus is the active site of production of rRNA, ribosomal rRNA. Okay, ribosomal RNA. Then question number 36. Which of the following is not a part of a transcription unit in DNA? Okay, promoter is a part, terminator is a part, destruction unit is a part, but inducer is not a part of the transcription unit in DNA. It is a protein. It is not a part. Okay, promoter region is present in a gene. Structural gene is present. This is the promoter gene. Okay. This is the terminator region. Okay, here we will be having poly A site. Okay. And you can use, you can say, row dependent and row independent termination method also. Okay, so that is known as the transcription unit. Okay, there is no inducer here. Now question number 37. Removal of RNA polymerase 3 from nucleoplasm will affect the synthesis of. Okay, very important question. Question number 37. So what thing will be affecting removal of RNA polymerase 3? So RNA polymerase 1 codes for what? RNA polymerase 1 codes for rRNA. That is what it does. And you can say type 2 is responsible for production of mRNA. Okay, and type 3 is responsible for production of tRNA. So type 3 RNA polymerase in case of eukaryotes produces tRNA. Okay. So that is the most important concept that you must be knowing. Okay. And one more thing I like to add to it. Okay. That a specific RNA polymerase codes for, you can say, this tRNA is coded by type 3. That is fine. Okay, that is all fine. But you have something known as RNA polymerase 1. Okay, RNA polymerase 1 also transcribes. Okay, there is a question in when it comes to RNA polymerase 1. I'll tell you a very hard question which came in NEET examination. So RNA polymerase 1 transcribes, I remember that question, eukaryotic ribosome. Which does not consist of. This question also came. Which does not consist of. Okay. So options were 28S. Okay. 5S rRNA. Then 5.8S. Okay, and 18S rRNA. Okay, these four options were there. So it was a very hard question. So we must know that eukaryote have 80S ribosomes and are made up of 60 plus 40. The large subunit, okay, is made up of, when you have 60 plus 40, 60S plus 40S. The large subunit is made up of 28S rRNA plus protein, then you have 5.8S and you have 5S and this one and plus proteins, and this one is 18S. Okay. So the large subunit 60 is made up of 28S RNA, I said 5.8S and 5S, and approximately you can say 45 proteins. Okay. Whereas the small subunit, the 40S, is comprised of, you can say 18S rRNA and approximately you can say 30 proteins. Okay. Now RNA polymerase gets attached to which one? Get attached to the smaller subunit of RNA during translation. Okay, during translation this question will be answered, that it gets attached to this one. Okay, so when it gets attached to the smaller subunit, okay, so what will happen is it will preclude this 5S rRNA. It will attach to this one and then later on polypeptide synthase will come. It will start from this one and it will preclude this 5S rRNA. It will not include this 5S rRNA. So this is a very, you can say, one of the hard questions which was asked in, you can say, NEET examination. Okay. So coming to the next question. What are the structures called that give an appearance as beads on a string in chromosomes when viewed under electron microscope? A very easy question, this one. Okay, beads on a string. 38th question. Okay, that is nucleosome. So we have learnt many times what nucleosome is. Okay, histone octamer, H2A, H2B, complete dimer, then H3, H and H4, single dimer. Okay, 146 to 148 base pair. Then I have described to you the chromatosome as well. Okay, so we'll be solving the remaining questions in the next class. Meanwhile, if you have had any kinds of doubts or queries, okay, any kind of doubts or queries, you can ask me your doubts and queries on my personal number, and I'll be replying as soon as possible. Okay, so one more thing I'll tell you, that the genes for, you can say, tRNA, I told you, is coded by RNA polymerase 3. Also, one thing noted down in this question, this 5S, okay, is also coded by RNA polymerase 3. Okay, RNA all 3, 5S rRNA. That is again the reason for this question's answer of it being 5S rRNA. It is also coded by RNA polymerase 3 along with tRNA. That's why RNA polymerase 1, during translation, it binds here and it precludes this, okay, because of this reason. Okay, so this was what got skipped. So this is the reason again. So 5S rRNA is coded by RNA polymerase 3. Always remember, that's why it gets precluded. Okay, so I hope you were able to comprehend the lecture, and let's meet in the next NEET class.